Scanning Electron Microscopy Validation Analysis by Brittany Fleck. So SEM EDX instrumentation. So SEM stands for Scanning Electron Microscopy, and EDX stands for Energy Dispersive X-ray Spectroscopy. So this entire instrument is placed into a vacuum where primary electrons are dispersed through an electron beam. So this electron beam is a highly focused and monochromatic beam that is sent through a series of multiple magnetic lenses. The SEM portion of this instrument detects the secondary X-rays and backscattered electrons. So backscattered electrons can visualize the various phases that are present in a sample's composition or general makeup. The secondary electrons visualize the sample's topography or the direct nature of the sample. EDX is an attached detector that is placed alongside the SEM detector. The way that EDX produces a signal is that it can determine an elemental composition or makeup through a two-step process. So the first step begins with energy being transferred to the atomic electron, which in turn is knocked off, leaving an empty hole in the sample, which you can see in figure 2, so the 1 and 2. And the next step fills the hole with another electron from a higher energy shell, and this movement will generate a X-ray that is read by the EDX detector. And electron signals are easily captured, and the elements then can be mapped and determine what elements are present within the sample. So SEM EDX microscopic and elemental analysis. So these techniques are a crucial tool for confirmatory analysis of forensic evidence. Sectioning off each of the instruments, so SEM is used to image the surface of a specimen at a very high magnification. So zooming in on those different surfaces of that sample. The SEM instrument is able to visualize the sample's surface morphology by giving details on its topography, composition, and crystallographic information. Now, separately, the EDX is an add-on to the SEM instrument and identifies the elemental information of that sample. So, SEM EDX sample types. This instrumentation is able to analyze different solids and liquids. Some of the different components that are able to be broken down are through its size, state, and conductive properties. So the sample size is able to vary as the setup is able to examine the various different sizes, so larger samples or portions of the sample, because the sample state is able to be a hard or soft sample, but there are different sampling preparations for each of them, so whether it's a solid or a liquid specimen. For SEM, it is necessary for the sample to be conducting in nature, so if it's not and it's like a non-metal, then it has to be coated to accommodate for its missing conductive properties for proper analysis using the instrument. Some examples that are seen in forensic evidence that can be analyzed using the SEM or EDX are biological samples, explosives, glass, GSR or gunshot residue, paint, soil, and toxins. So SEM EDX sampling methods. So these different, within these sampling methods, the sampling condition must be very clean and dry. Proper sampling preparation allows for the best image clarity and detail of the sample. So cross sections of the sample are then fixed by slicing, acid etching, or abrasions. So sampling methods can range between the different types of samples, so whether it is a solid, liquid, or its surface has conducting properties. So for non-conducting samples, its surface needs to be coated with a conducting material such as carbon, gold, or platinum. In SEM EDX, metallic stubs are used for various types of samples, and these are made of beryllium or pyrolytic carbon. For liquid samples, these are coated and dried on the stub within the instrument, and fixation is often used when the sample needs to be treated with a buffered chemical treatment, such as glutaraldehyde, in order to preserve or stabilize its sample's structure. Skipping the fixation process can lead to sample damage during the dehydration, which follows after fixation steps. So the dehydration step is generally done using a critical point drying or freeze drying in order to produce those highest quality results. For solid samples, these are a little bit easier types of samples, and a small sample is scattered on an adhesive tape. So the study that I chose was a recent study that analyzed the varying lines of growth of cementum on erupted or raised permanent teeth of women who had given birth to a child. So cementum is a hard, like calcified layer of tissue that covers the root of the tooth. And so this is what they focused on for their sample types. So SEM EDX use in the study. So 25 teeth from females who had been through at least one pregnancy were taken from. The female donors were between the ages of 18 to 76, so they had a wide range of individuals, and each of the donors were prompted to take a survey to gain a general basic information such as their age, the number of childbirths, and their current general health conditions. So SEM EDX was used for both microscopic and elemental analysis. Specifically for the microscopic analysis, samples were imaged measuring the varying thicknesses of the incremental lines on each of the tooth samples that were collected. And then as for the elemental analysis, the study used X-ray peaks to determine the variation in the varying elemental levels for each of these samples. So this had shown that there was a drastic increase in calcium and phosphorus in those women who had given birth. So in the study, as for the samples used in sampling methods, the samples were tooth samples that were collected from female donors, and each of the sample was cut into transversal sections. And then as for the sampling methods, each of the samples were preserved in a formalin until their thermal and UV sterilization was able to be performed on each of the samples. So the sliced tooth samples were then rinsed with ethanol, and then they were wet rubbed with cotton buds. So after that preparation, each sample was attached to an aluminum stub on the SEM EDX instrument to create that conductive property needed for analysis. And so on the evaluation of the results and conclusions, so as for the results of the study, it was only 6 of the teeth samples were used for further examination. Each of the samples were separated based on those varying elemental content based on early age, later age, or dentin region. So this is going to be those varying ages. So if we kind of jump back, it was ages between 18 to roughly 76 years old. And so the authors noticed that there was an increase in calcium, phosphorus, and oxygen. And then as for the overall conclusion, um, it was found that pregnancy that resulted in a childbirth was able to determine was able to be determined using this SEM EDX elemental analysis along the line of growth on the dental cementum. So the evaluation of the study and some forensic applications. So cementum, as mentioned before, is deposited continuously throughout a person's life. So it alone is able to determine general age of an individual. So this can be used for mass, like mass destruction, or when a lot of people have been killed, they can range the different ages of each of those individuals. And so with forensic odontology, it uses dental evidence or tooth samples, which are able to determine valuable information from these individuals, even multiple or several years after their death. And so the purpose of this study and why I chose it was it used the confirmatory analysis of SEM EDX in order to narrow down specific victims or suspect searches and identification by determining their age, estimation, and other vital details from unidentified people. So if they find the goal of the researchers was basically to find a overall like if they find a buried body, they're able to determine the age estimation, whether they're female, male, and then if they have given birth to a child. And so the successes slash improvements for this study, as far as the successes, I think that the study was very thorough and in depth with the qualitative and semi quantitative or fingerprinting of the elemental analysis. I thought that that was pretty thorough. They even surveyed their participants to get a general idea or overview on who they were working with and the unique traumas that each of the donors had been through. And the researchers performed the proper sampling methods for each of the sample types or tooth samples that were used. Some two improvements that I noticed throughout the study was that they even admitted that they could have been more in depth with their surveys, such as clarifying the difference between an actual childbirth, abortion, stillborn, miscarriage. So that may have a factor in the different levels of the cementum in the tooth samples. And then another one was the researchers could have taken images of each annulation of the teeth samples on the SEM scans and assigned a specific age to it. So those were just some of the improvements that I noticed. And so these are my references for my information, and then this is the references for my photos. Thank you.